This is the last video for chapters 25 and 26 PowerPoint, and we left off with the last one on looking at how different cladograms or phylogenetic trees can show different things. And some of them, the branch lengths can indicate the time that has passed between the most common distant ancestor and the actual divergence of the different organisms that you see here. So thinking about time, the next topic I want to talk to you about is molecular clocks and how they can help track evolutionary time. So in the recent decades, evolutionary biologists have been investigating the possibility that some evolutionary changes occur in a clock-like fashion. So over the course of millions of years, mutations can build up in any given stretch of DNA at what they believe is a reliable rate. For example, the gene that codes for the protein alpha-globin, which is part of hemoglobin, has had base changes at a rate of about 0.56 changes per base pair per billion years. So if we have a rate like this that's reliable, that gene can now be used as a molecular clock. So in essence, a molecular clock uses constant rates of evolution in some genes to estimate the absolute time of evolutionary change. So this brings up the terms that we introduced earlier, the orthologous genes, which again I'm sure I'm saying that wrong, which have nucleotide substitutions proportional to the time since they last shared a common ancestor, and the paralogous genes, which have nucleotide substitutions proportional to the time since the genes became duplicated. So the orthologous genes, the first type, are the ones that there's only the complete set. There's not a duplicate set present of the genes. In the paralogous genes, you have duplications that have occurred over time. So the problem with the molecular clock theory is that the molecular clock does not necessarily run as smoothly as expected. For example, if you have neutral mutations, such as a substitution, irregularities from natural selection can also favor DNA changes, even if the mutations are not neutral. Natural selection may favor some over others. So estimates of evolutionary divergence older than the fossil record have a high degree of uncertainty, though the use of multiple genes or genes can improve the estimates. So the more that we can use in relative comparison is if we're using these as a clock or as a standard, obviously the more that we have, the better chances they'll be accurate. Another concept that we need to talk about a little bit is the difference between horizontal and lateral gene transfer. So lateral gene transfer is as we pass genes down the line through ancestors to descendants. That's lateral gene transfer. But horizontal gene transfer is the movement of genes from one genome or one species to another. And sometimes this can be entirely different kingdoms in terms of the horizontal gene transfer. For example, from bacteria to eukaryotic eukarya. So the tree of life suggests that eukaryotes and archaea, for example, are more closely related to each other than to bacteria. And that means archaea to bacteria, there's less relationship or close relationship than archaea to eukaryotes. So the tree of life is based largely on ribosomal RNA genes, which again are the ones that oh gosh, this silly thing, which are the ones that change very slowly over time. So those are the ones that are used for the long-term relationships that we're looking at. And there have been substantial exchanges of genes between organisms in different domains. So this horizontal gene transfer occurs by exchange of transposable elements between the exchange of plasmids through the using viruses as vectors, viral infections, and also through the fusion of different organisms. And we know again that there has been transfer between species, like between prokaryotes and eukaryotes, and between the 3 DNA-containing organelles of eukaryotes. So there can be gene transfer between the nucleus, the DNA in the nucleus, between the DNA in the mitochondria, and between the DNA of the chloroplasts and that of the nucleus. The acquisition of DNA through horizontal gene transfer is distinguished from the transmission of genetic material from parents to offspring during reproduction, which again is the vertical gene transfer. So when we find disparities between the phylogenetic trees that are showing the relationship of genes, those disparities can be explained by this horizontal gene transfer. And this is just a picture of a Mediterranean gecko, but this is one organism that has actually been proven to have horizontally transferred genes within it. So this is, there are 2 last things to leave you with. One of them is that because of this horizontal gene transfer being so common, there are some biologists that think that the early history of life really looks a lot more like this than as straight lines of descent in a nicely branching tree. That it gets pretty convoluted when we actually look at the DNA and look at molecular data. And this has to do with a lot of horizontal gene transfer. And then the final thing to leave you with is just looking again and thinking back to where we started with chapter 25 and looking at the geologic record. So as we're looking at phylogeny and looking at the relationship of organisms, it helps to put it into the perspective of what has occurred over geologic time. So this animation will review that for you.
By studying various fossil records, geologists have put together a geologic record. Evidence in the fossil record of large-scale extinctions and/or enormous radiation of many diverse new life forms defines the main boundaries within the geologic record. As you move through the geologic record, the names and dates will change. Also, a diagram at the upper left will show what is known of the history of continental drift, based on evidence dating from the Mesozoic era. During the Precambrian time, which began at least 4,600 million years ago and ended 542 million years ago, life began on Earth. The first prokaryotic cells and cyanobacteria appeared, as well as the first animals. The beginning of the Paleozoic era, 542 million years ago, marked the origin of most modern animal phyla. The first plants, jawless fish, bony fish, and amphibians evolved. Amphibians became the dominant vertebrate life form, while the first reptiles and seed plants appeared. By the end of the era, 251 million years ago, many marine and terrestrial organisms had become extinct. The Mesozoic era stretched from 251 million to 65 million years ago. At the beginning of this era, the reptiles radiated out as dinosaurs to dominate the landscape, along with the cone-bearing plants, or gymnosperms.